Right. So the next thing we are going to look at is the functions of proteins. Now, proteins aid in movement. They are important parts of hormones and they are enzymes. That's the reason why you have this saying that goes that all enzymes are protein, but not all proteins are enzymes. They are parts of hemoglobin, myoglobin, which are transport protein for transportation of oxygen and nutrients in the body. Proteins help in growth and differentiation. Then the transmission of nerve impulses, like the neurotransmitters. There are neurotransmitters that are proteins. And then you have in vision. All right. All these are some of the functions of protein. This is just something you can just memorize, and then you reproduce it in the exam. No more explanation. Now, classification of proteins. Now, when we talk about the classification of proteins, what are we looking at? Proteins are classified based on various criteria. Proteins are not just classified based on one criteria. Now, in the exam, you might be given, because this is one question that will definitely, if there's a question that will come out under this one, is this classification of protein, right? They will always ask you to classify proteins. Now, if they say classify protein, it means you classify them based on the different criteria, and they give example and explain if possible. However, if you are not asked to classify, if they just give you classify based on solubility or classify based on biological function, then you just have to go straight and classify based on biological function. That means the criteria you are asked, or the criterion you are asked to classify those proteins, is what? Is based on the biological functions of those proteins. So classification of protein, we have the complete or first class protein. Now, those complete or first class protein, they contain all essential amino acid. Now, you are told that we have 10 essential amino acid and what? 10 non-essential amino acid. Some people say 11 essential, 9 non-essential, and some. But you just know that we have just around this region, right? So, any protein that contains all the essential amino acids is a complete protein. Now, what are the essential amino acids? Those are those amino acids that your body cannot manufacture, hence you have to take them in as your meal. Is that okay? Those ones, they are what? Essential amino acids. They are essential to your growth and development, but your body cannot actually manufacture them, hence you have to take them in as your meal. Example is egg, milk, meat, fish, yeast, and soybean. All those ones, they fall under complete protein. So if you are eating egg, you are eating a complete protein. Milk, complete protein. Meat, complete protein. Fish, complete protein. Now, incomplete or second class proteins. The first one is complete or first class. Second one is incomplete or second class protein. They lack one or more essential amino acid, e.g., the cereals, the legumes, and then nuts. For instance, your pea, now, they lack methionine. Is an amino acid. So any pea you see, most peas you see, they lack what? Methionine as the essential amino acid. So most times you usually add them with other proteins that contain that amino acid that they are missing. For instance, you can add pea with corn. Corn lacks lysine and tryptophan. Is that okay? You can browse it on the internet. Corn lacks lysine and tryptophan. Pea lacks methionine, but corn has methionine. Corn lacks lysine and tryptophan, but pea has lysine and tryptophan. So you combine them to get a complete protein diet. Is that okay? Then the other one is rice. Rice is not a complete protein. Do you know that rice is a protein? Yes, but it's not a complete protein because rice lacks lysine. Are you getting it now? That's the reason why you now eat rice and beans. So when you are eating rice and beans, you are eating a complete protein because beans contains methionine and tryptophan. Is that okay? With that one. Now, the next one we are going to look at is classification based on biological functions of proteins. Based on what? Biological function. Now, this one is your exam question. it usually comes out. They usually ask, with example, classify proteins based on function. You can see the exam question here. With example, classify protein based on what? Based on function. So when we talk about classification based on functions, I told you we listed the functions of proteins here: movement, hormones, enzymes, hemoglobin, growth, differentiation, transmit nerve impulse, and vision. All right. So when we classify proteins based on function, you have the functional class that we are listed here. Functional classes are these things you see here: enzymes, regulatory proteins, transport proteins, storage proteins, contractile and motile proteins, structural proteins. So you take all these ones now, write them out, keep them. If it comes out in the exam, you just attack. It's one question on its own. Is that okay? So proteins are enzymes are protein, but not all proteins are enzyme. What are enzymes? We say enzymes are organic catalysts, proteinous in nature. That help to speed up the rate of chemical reaction without being used up during the course of the what reaction. Example of enzymes. I can list as much examples of enzymes as possible because of the fact that I'm in the medical field, and enzymes actions are very, very, very important in the functioning of drugs. Talking about the pharmacokinetics of drugs, enzymes are very, very important. All right. So we have trypsin, we have phosphofructokinase, which is an enzyme that is very, very important during the metabolism of glucose. We have alcohol dehydrogenase. This is an oxidoreductase enzyme. We have the catalase. We have the malic enzyme. All these are the examples of what enzymes: phosphofructokinase, trypsin, even your normal ptyalin, the one in your mouth, amylase, lactase. You can list all those on their enzyme too. Lactase, maltase that break down lactose, maltase that break down maltose, sucrase that break down sucrose. You can list all those enzymes. Hexokinase, glucokinase. Those on their what? Their enzymes. There are many enzymes that you should know. You just bring the knowledge of everything you've learned about digestion in your secondary school. The enzymes that function in the various processes of digestion, trypsin, chemotrypsin, and so on and so forth. Then we have the regulatory proteins. Regulatory proteins are proteins that regulate one or two metabolic processes in the body. Example is insulin. I know that a dysfunctionality, or let's say decreased secretion of insulin, or inability of insulin to carry out its function very well, it causes diabetes, and that's why we say diabetes is a metabolic disorder. Is that okay? We have insulin. These ones are regulatory proteins. Insulin is an hormone. Most regulatory proteins are hormone. We have somatotropin. Somatotropin is growth hormone. Thyrotropin, from thyroid. Then you have lac repressor. And then transport proteins. Under transport proteins, you have hemoglobin, which transport oxygen via, when it combines with oxygen, it forms oxyhemoglobin and transport oxygen through and forth the body. And then we have the serum albumin and the glucose transporters. These glucose transporters, they are proteins that help to transport glucose across the cell membrane. We have different types of glucose transporter, I believe from GLUT1 to GLUT7 also. They are called GLUT. They transport glucose to different parts of the body, across the cell membrane in different parts of the body. You have GLUT1, you have GLUT2, GLUT3, GLUT4. I think you have GLUT. There are 6, but you don't have GLUT6, but you have GLUT7, something like that, if I can still remember very well. Those are called glucose transporters. Now, for those of you in sciences, biological sciences, you may learn this. Biochemistry, you will definitely learn this. And then chemistry, this might just be the last time you are going to see something like this, because remember, during my stay in chemistry, I never came across anything like glucose transporters. But in pharmacy, you are going to see it a lot. All right, so we have storage proteins like ovalbumin, casein, and ferritin. Contractile and motile proteins. Those are the proteins that are present in what? In muscle tissues. All right, because you say muscle tissue does what? They contract. So contractile and motor proteins are found in muscle tissue. Example is actin, myosin. Specifically, if you name these two, because they are going to tell you to give examples and classify proteins according to biological functions, and then give example. If you can give two examples each, you can give three examples each, you are very, very good. Like this one, if they talk about contractile and motor proteins, actin and myosin must always be the first one you are going to look at. Then tubulin, dynein, and what? Kinesin. And then structural proteins. Under structural proteins, you have the alpha carotene, collagen, elastin, fibrin, proteoglycan. Proteoglycans, they are found in the cell walls of some insects, right? You find them in the cell wall of some insects, and then which are also called glycoproteins. And alpha carotene, they are the ones that are found in your hair. Collagen, bone tissues. Are you getting it? Alpha carotene, the proteins that makes your hair to stand, is collagen. In your hairs and your nail, you have alpha keratin rather. Is that okay? And then scaffold proteins. To be honest with you, I have no idea of what the scaffold protein is all about. So we can ask ChatGPT to give us details of what scaffold protein is. ChatGPT, so this is part of the learning process. You have scaffold protein, what they are for. Scaffold protein and structural molecule that play a role in organizing. Okay, still coming up. Okay. Now, you can see here that the scaffold proteins are structural proteins that play a role in organizing and coordinating cellular signaling pathway. They act as platform or framework that bring together various signaling molecules such as enzymes, receptors, and other proteins, facilitating their interaction and ensuring the efficiency and specificity of the signaling cascade. The term scaffold protein is usually used in the context of intracellular signaling. And to be honest with you, I still don't really understand what they want here. All right, but maybe this one is for advanced level studies, but you can just know it and you are good to go. All right, so that is that. You have the GROB2, CROK, Christian religious knowledge, SHC, statistics, STAT, and IRS1. Now, protective and exploitative proteins. We have the immunoglobulins for immune system, thrombin, thrombocyte, white blood thrombocytes, right? Blood platelets, that's thrombin. Fibrinogen, that produces fibrin in blood clotting. Antifreeze protein. Then we have snake and bee venom protein, diphtheria toxin, and ricin. All these ones are examples of what protein. Then we have the exotic proteins like monelin, resilin, glue proteins. All these ones are things you can just read, memorize, and go and write your exam and score your very good A. Now, classification based on shape and solubility. Now, based on shape and solubility, we have fibrous proteins. Then you have after fibrous, we have globular, and then we have membrane protein based on shape and solubility. Now, what are fibrous proteins? Fibrous proteins, they are simple, thin, fiber-like shape and regular linear structure. They have this regular linear structure. They are simple, they are thin, they are fiber-like shape, and have the regular linear structure. Fibrous protein play an important role in providing structural rigidity and in contractile movement, like the ones in the muscles, actin and myosin. They are insoluble in water or in dilute salt solution. So when you talk about the solubility of fibrous protein, in water they are insoluble, even in dilute salt solution they are insoluble. Now there are two types of fibrous proteins, which are alpha and beta keratin. The alpha keratin are the protein that make up your hair, wool, skin, and nails. So when you check your hair, that protein that is actually present there is what? Alpha keratin. Even your skin, proteins there, alpha keratin. Your nails, the protein there, alpha keratin. Then we have the beta keratins. They are proteins that are found in the feathers of birds and the scales of reptiles. Those ones, they are looking a little bit harder, right? They are found in the feathers of birds and the scales of reptiles. So this one, you can actually find them in animal, while this one for human, the alpha keratin for human. Now, in beta carotene, the protein consists of large amount of beta-plated sheet structure. You've been taught the structures of protein. I think I actually did a little bit review on the structures of protein. We have the alpha helix, and then we have the beta-plated sheet. All right. So beta carotenes are the ones that have this beta plated structure of protein. E.g., told you myosin, which are found in the muscles. Also, we have F-actin, we have keractin, we have collagen, elastin, tubulin, and what? Spectrin. These are examples of fibrous protein. I just, but most times I don't usually ask questions on that fibrous protein. Classification based on shape and solubilities, based on biological functions you are going to see. But just know it because your time may be different. All right. So we have the fibrous protein. This is an example of the fibrous protein. Now, most fibrous protein, they lack the 3D structure or the quaternary structure of protein. They are usually primary or secondary structure. Now, you are going to learn about the primary or secondary structures of protein, maybe in our next lecture, or I think it's in the first video I sent on that protein. The 3D structures of protein. We have the primary structure of protein, which contains the amino acid, the order of the amino acid, and is a linear shape protein. Are you getting it? Then when the protein becomes too long, it begins to fold to give us the 3D structure. You can see that this one is a linear structure. Are you getting it? That is where you now have secondary structure, where you have the alpha helix and beta helix. All those ones, they fall under fibrous protein. So fibrous protein is more like secondary protein structure, like the alpha helix and beta helix, like the ones you can see here. Then for the globular proteins, they have the 3D and the quaternary structure. Now, a protein, when the protein is growing, you first of all, not growing per se, when you have a protein, the first set of amino acid sequence give the linear structure of the protein, which is the primary structure. Now, as time goes on, that linear structure will begin to fold a little, which gives the secondary structure, which now give the alpha helix and beta helix. As time goes on, this secondary structure will now fold. It gets too long to now fold like this to give us the 3D structure, which you can see here. Are you getting it? So this is globular proteins are mainly 3D structures of protein. The globular proteins are spherical in shape. The polypeptide chain is compactly folded. Emphasis, they are folded compactly so that the hydrophobic amino acid side chains are in the interior of the molecule and the hydrophilic side chain are on the outside, exposed to the solvent. Are you getting it now? Now, the hydrophobic amino acid side chain. When you talk about hydrophobic amino acid side chains, there are some amino acids like valine, like leucine, like isoleucine. Those ones, they have hydrophobic because they are made up of alkyl group as part of their side chains. Those ones, they are hydrophobic. They are not water soluble. But when you now come to glutamate, threonine, methionine, all those ones that have polar side chains, are you getting it? Those ones, they are hydrophilic. Now, a protein, we have hydrophobic group as their side chain, and the protein will also have hydrophilic group as their side chain. If you have such kind of a protein, what will happen is that when they want to form globular protein, they will fold in such a way that the hydrophobic are on the interior. So if this is a spherical structure, the hydrophobic side chains, all those ones that have hydrophobic side chain, will be on the interior, while those ones that have the hydrophilic side chains will be on the exterior part of the spherical structure. Is that okay? Exactly. Now, globular proteins, they function as enzymes, chemical signaling compound, transporters of other compounds, and antibiotics like hemoglobin, myoglobin, albumin, immunoglobulin, pepsin, lysozymes, and amylase. All right. We did an expository study of immunoglobulin, I think last semester, and we also talked about the antibiotics and different class of antibiotics in pharmacy school. But you guys don't need all this, just memorize because if you want to go into detail and start explaining, you can spend hours explaining only what immunoglobulin is. So just know it as per figure-headed knowledge that this is it, but not in-depth understanding of what it's actually about. Then we have the membrane protein. I've actually explained this membrane protein before now. Told you that the cell membrane is comprised of three important things. It's comprised of lipids, which are phospholipid, cholesterol, and glycolipid. consists of proteins, integral proteins, and the peripheral proteins. And then we talked about the carbohydrate moiety present, but not that much carbohydrate, all right, that are present in them. So the membrane proteins of the cell. Now, the membrane protein functions as enzyme, cell receptors, especially cell receptors. They functions as enzyme, they functions as cell receptors and transporters of compound in and out of the cell. I've explained that before. Those that transport compounds out in and out of the cell, they are called channels or carriers. Are you getting it? Channels or protein carriers. Now, membrane proteins are insoluble in aqueous solution, but can be solubilized in solution of detergent. The reason why they are insoluble in aqueous solution is because these membrane proteins, they are actually suspended in the lipid layer, so they have to be soluble in the what? In the lipid layer. But when you now take them to aqueous layer, they are what? Very, very insoluble. Now, membrane proteins have fewer hydrophilic amino acid than cytosolic protein. Now, when we talk about having fewer hydrophilic amino acid, hydrophilic means what? Water-loving amino acid. True or false? Now, because they have fewer water-loving amino acid, that is the reason why they are insoluble in aqueous solution. Example is aquaporine. Aquaporine, they are called special water channels. Aquaporines, as the name implies, aqua, water, pour, where water actually gets into the cell. Are you getting it? That's what we call the what? Aquaporines. So aquaporines are special water channel. And then we have the glucose transporters. Like we said, GLUT. Glucose is so large that it cannot just swim across the membrane via the concentration gradient. Or are you getting it? So what actually happens is that they use special transporters to transport glucose into the cell. I will have them called the glucose transporter. Now the glucose transporter will have GLUT. I think GLUT, there are 6 of them, GLUT 1 to GLUT 7, but there is no GLUT 6. I believe so. Let's confirm, because I've said this thing twice so that it will not become a knowledge in your head that this is what Sami said that glucose transporters are. So let's check glucose, right? Transporters. Say glucose transporter and protein that facilitate transport of glucose across the cell membrane, of course, which I have stated before. Okay, there are 6 of them. This 9, this 9, 1, 9, 10. Don't reason them. There are 6 that are very, very important to us, which are GLUT 1 to GLUT 7, but there is no GLUT 6. So I'm actually very, very correct. Are you getting it?